Thank you very much, James. Again, my name is Jose, and I've been in technical support for a while. Today we'll be talking about strands and strand definitions as found in our manifests and in other places, and how and why we need to know and be familiar with these strand designations and genotype designations. So today, my objective for you all is to understand, number one, Illumina's top bot strand designation and genotype A, genotype B allele designations. I want you to be able to identify easily a given SNP allele and surrounding sequence as either top or bot strand, and which bases correspond to allele A and allele B. Also, I want to define other strand designations contained in Infinium genotyping manifests and understand how these relate to various genotyping report formats. And finally, I want you to be able to convert a report from one strand format to another strand format using the top bot designations of each of the formats. And the reason we want to do this and to be able to do this is because historically, genotyping data have been reported based on a wide variety of strand designations. Most current analysis tools require alleles to be derived from the reference strand, or forward strand, or plus strand, also known. The data generated using other strand designations must be convertible to the reference strand or to any particular strand of interest that you're using, depending on the software that you're using to do the analysis. All right, so where are these strand designations found? Let's take a look at an Illumina BeadChip manifest. Now, as you know, the manifest is essentially a list of SNP assays for a particular given BeadChip type, all right? And it also contains the associated codes and some genomic annotations. So here's the manifest. There is a link below to a recorded webinar that goes into what a manifest is and how it works in the context of Illumina scanning and data analysis. So the link is included. Where else can we find these strand designations? We can find them in a Genome Studio project. So once you've scanned the chip and entered it into a Genome Studio analysis project, you'll see that the alleles, the data itself, is reported in potentially various different strand designations. By default, a genotype in a Genome Studio project is reported as a genotype A, genotype B, but there are other available views, and you can choose to view them through the Genome Studio menus. All right. One key thing to note is that depending on which strand designation you're choosing, the allele bases that you see may be different. You can also see the strand designations in the reports that are output by Genome Studio and by various other third-party software that many scientists use to analyze the data. So in these reports, you can see similarly that depending on which allele, depending on which strand designation you choose to see, you may see the different alleles. Now, in order to understand how these different strand designations are related and how we can reconcile them, we need to understand how and why the different strand designations came about in the first place. So we will be exploring the history of these strand designations. Let's start with the Human Genome Project. I'm sure we're all fairly familiar with what the project is and what its goal was. The project was an international research project with the goal of determining the sequence of nucleotide base pairs that make up the human DNA, and of identifying and mapping all the genes of the human genome. The final assembled sequence is a combined mosaic of a small number of anonymous donors, all of them of European origin. So the time frame of this project was essentially the decade of the 1990s. It was completed in the mid-2000s. Now, as you know, a chromosome is a long piece of DNA, a single molecule of DNA. Like all DNA molecules, the chromosome is bidirectional. That is, genes are transcribed from both strands in both directions, and each strand is of equal importance. Now, the goal of the Human Genome Project was to create a reference sequence. Now, in order to do that, they had to choose one of the two strands of each chromosome as the reference strand. And again, since both strands were equally important, we needed to choose a structure by which we could select the reference strand. Fortunately, all chromosomes have a structure called a centromere. The centromere divides a chromosome into an asymmetrical molecule, a short arm, which was designated a P arm, and a long arm, which is designated a Q arm. Now, the strand running 5' to 3' from the P to the Q strand was chosen as the plus or reference strand. The strand running opposite direction, 5' to 3' from Q to P, has been designated the minus or negative strand. Unfortunately, for our purposes, because as with all new things in science, many different standards were initially adopted. So the plus strand was also alternatively known as the reference strand or the forward strand, while the minus strand came to be known as the alternate strand or reverse strand. Now, as the Human Genome Project was going along, a couple of other projects developed. The HapMap project was an organization that aimed to develop a haplotype map of the human genome to describe common patterns of human genetic variation. The HapMap was used to find genetic variants affecting health, disease, and responses to drugs, and environmental factors. Another project is the SNP database, a single nucleotide polymorphism database, or dbSNP. It's a free public archive for genetic variation within and across different species, developed and hosted by the NCBI. Now, what's important to note about these different projects that cataloged variations is that they decided to treat the strandedness of their entries in different ways. Now, dbSNP accepted entries from submitters regardless of the strand assignment, because in the beginning there was no consensus genomic sequence. This means that the SNP and surrounding sequences that were submitted by scientists could either be on the plus or the minus strand. HapMap chose to designate the P to Q strand as a forward strand and the Q to P as reverse rather than the plus or minus. And HapMap attempted to catalog their variations based on the forward strand initially. Because of the state of flux of the Human Genome Project, entries were inconsistently cataloged. Now, there's been a more recent project called the 1,000 Genomes Project. Now, they have archived all their sequences in the genomic forward plus strand. It was begun after the human genome was completed. As Illumina started to create their SNP assays and their arrays, Illumina chose dbSNP as its primary source for SNP sequences and assay design. But now, because dbSNP entries did not initially include genomic plus and minus designations, the Illumina manifests did not include that information either. To report genotype alleles, Illumina chose to use the dbSNP entries and, in general, to designate source sequences as forward sequences. By definition, Illumina forward alleles and sequences equals the SNP and surrounding sequence as it exists in the source from which the sequence was derived. So Illumina forward is equal to the sequence as it exists from the source. Now, when Genome Studio generates a forward strand report, the alleles are reported and based on the alleles as they exist in the source strand. So in the manifest, the column source seq lists the sequence of the SNP and surrounding area as derived from the source that Illumina derived the SNP information from. This means what this means ultimately is that Illumina's forward is different from the forward strand of the genome. Okay, so let's take a look at these strand designations as we have them so far. The reference or plus strand of the genome is the P to Q 5 to 3' strand, and the genome coordinates are based on that strand. The strand is also known as the forward strand of the genome and also known as the reference strand. Now, dbSNP entries did not initially assign their sequences that were entered to either plus or minus because that information was not in existence at the time. Illumina based its SNP assays on dbSNP entries and designated those entries pulled from dbSNP as the forward strand. And the bottom line is that Illumina's forward strand is not equal to the genomic forward strand. And that, of course, has its many consequences. Now that we've looked at the existing standardized strand designations for the genome and how they relate to Illumina's designations, let's look at Illumina's top bot strand designation and allele A and allele B assignments. And why did Illumina feel this was an important thing to develop? Well, there was a need for a more stable strand designation at the time that would not switch every time the human genome sequence was updated and modified. Eventually, upon completion of the Human Genome Project, we expected a standard of reporting alleles based on the plus or forward or reference strand of the genome to be adopted. Now, a means to compare, associate, and or convert other reports, older reports based on the forward or other strand designations to the accepted standard would be useful. And the sequence context-based strand designation would be very stable because SNP and surrounding sequences we don't expect to change. Thus, Illumina decided to create a different strand designation and to assign allele A and allele B base designations. And so we invented the top and bot strand designations and the A and B allele designations. Let's take a look at how this strand designation and allele designation system works. Let's start with what I will call class number 1 of SNPs. These are non-AT and non-CG SNPs, and you'll see why we divide these into two different classes as we go along. We begin by designating a definition of determinant bases. Bases A and T are designated by definition as determinant bases. Now, for non-AT and non-CG SNPs, in order to designate genotype A and genotype B, we simply say that A and T bases are genotype A and C and G bases are genotype B. So, also for the strands, the strand which contains the A allele is the top strand. The strand which contains the T allele is the bot strand. And here's a simple table looking at some SNPs and how we use the SNP bases to designate strand and allele. So it's an AG SNP. For that particular strand, it is the top strand, and allele A and allele B are A and G respectively. For AC, similarly, allele A and allele B are A and C respectively, and since it's base A, it's the top strand. For the bot strand, we have the T and G and T and C, with the T's designating the strand as bot and also as allele A, and C and G, of course, as allele B. It's a relatively simple system for non-AT and non-CG SNPs. Let's take a look at a few examples. Now, I've put here some double-stranded representations of SNPs with the A and G and T and C bases. And let's take a few seconds and see if you can yourselves assign top and bot strands for each of these SNPs. I'll give you about 30 seconds. All right, let's take a look and see if you got these right. So for the top and bot strand designations, you can see that all the strands which contain A's have been assigned to the top as top strand, and all of the strands with the T's have been assigned bot strands. Now let's look at genotype A and genotype B. I'll give you a few seconds to fill the table in and take a look. All right, let's take a look at what the answers are here. You can see that for genotype A, for this class of SNP, all of the A's and T's are genotype A, and all the C's and G's are genotype B. So for the first strand in AG, the A designates that strand as the top, genotype A is A, genotype B is G. And you can see likewise for the other strand, because it has the T, it's designated as the bot strand, and genotype A and genotype B are again correspondingly T and C. And you can carry this logic all the way through for non-AT and non-CG SNPs. If you look at a manifest, you can see here for this SNP example, you have RS10000073. The Illumina, if you look at this SNP again, in the dbSNP entry, you see the sequence that is entered into dbSNP. It contains a CT SNP designation. So this SNP, as entered in dbSNP, because it has the T allele in the strand designation, the strand designation is bot as it is entered in dbSNP. Allele A is T, allele B is C. So that's a look at a SNP as it's entered in dbSNP. Now let's switch gears and look at class 2 SNPs. Now these are AT and CG SNPs. Because an AT SNP has A and T bases on both strands, we cannot use A and T directly to determine the top and bot strands or the A and B alleles. Similarly for C and G, because the same bases are existent in both strands, we cannot use the same rules as class 1. So we've adopted another set of procedures, rules to assign the strands and the alleles. The first step is to assign the strands using, again, A and T as determinant bases. Now, in the flanking sequences on either side of the SNP, you need to locate the closest determinant bases on either side of the SNP. That is, where is the closest A or T to the SNP? Now, if the determinant bases are equidistant, in other words, you have an A or T right next to the SNP on both sides, then you need to look further out. And you need to continue to look further out until you can find a determinant base, A or T, which is standing alone with no other partner on the other side. So the strand in which that determinant base is 5' of the SNP is the top strand. The strand which the determinant base is 3' of the SNP is the bot strand. This is how we assign strands for AT and CG SNPs. Now to assign the genotypes, once these top and bot assignments have been made, we simply look at the bases, and on the top strand, genotype A and genotype B are what I call alphabetical. And the bot strand, genotype A and genotype B are anti-alphabetical. So it'll become clear when we start looking at some examples. All right. Now, let's take a look at a few AT and CG SNPs. I've added some surrounding sequence so that we can do the strand determination. In the first case, we have a CGA followed by an AT SNP and then a GTC. If you look at that strand and you look outward for a determinant base, there is a determinant base immediately adjacent to the SNP. It is on the 5' end of that upper strand. So the upper strand is designated as the top strand. Similarly, if the other strand is top, then this strand must be the bot strand, the lower strand. And you can see that the T, which is corresponding to that A, is 3' of the SNP position on that strand. Let's go through the other SNPs. You can see here in this example, the upper strand is a bot strand because the closest determinant base is 3' of the SNP. If you look at the other strand, the corresponding A to the T, the corresponding A is 5' of that SNP, and so it is the top strand. For the next two, let's take a few seconds and see if we can determine those. I'll give you a few seconds to figure it out, and then we'll look and see if you've got it right. All right, let's see. Here you can see that we have a bot strand on the upper strand and a top strand on the lower strand. In this upper strand, you look on either side of the SNP and you see that there's an equidistant determinant base, so we have to look further out. And so if you look at the further out base, we see that the next determinant base is in the second position away from the SNP. In the upper strand case, you can see that it happens to be 3' of the SNP, so this is a bot strand. The corresponding determinant base on the lower strand is 5', and that makes it a top strand. For the next SNP, similarly, you look for the determinant bases, and in this case, you have to go all the way out to the third position to find a determinant base that's not ambiguous. So the upper strand is top and the lower strand is bot in this particular case. Now, as I was talking about genotype A and genotype B, remember that for top strand, the bases are alphabetical, and for the bot strand, the bases are anti-alphabetical. So let's see what I mean. In the first example, the top strand has an AT, this is an AT SNP. The top strand will be alphabetical. A comes before T in the alphabet, so the top strand, genotype A is A, and genotype B is T. In the bot strand, genotype A is T, and genotype B is A. Let's take a look at the next one. And this is an AT SNP. Again, the top strand, which is the lower strand, is alphabetical. Genotype A is A, genotype B is T. The bot strand is anti-alphabetical. Genotype A is T, genotype B is A. Similarly, for the CG strand, the CG alleles, I'll give you a few seconds to fill that one in. All right. Again, top strand is always going to be alphabetical for these SNPs, and C comes before G. So on the top strand, allele A is C, allele B is G. For the bot strand, allele A is G, and allele B is C. And for the others, the last SNP, top and bot are upper and lower. So for the upper strand, C is genotype A and G is genotype B, and G is genotype A and C is genotype B for the bot strand. Now, to review, top and bot and allele A and allele B were invented by Illumina as a response to the instability of the strand designations for SNPs relative to the plus and minus strand of the genome. Top and bot strand designations are intended as a reference strand designation to compare Illumina forward and other strand designations to each other and to the plus and minus or forward and reverse strands of the genome. Top and bot and allele A and allele B are based on sequence context and therefore are very stable. Now that we've got an idea of existing strand designations and Illumina's top bot strand designations and allele A, allele B alleles designations, let's take a look at the strand designations in the manifest and why these strand designations are important. Now first, let's look at something in the manifest called the Illumina Strand, also known as the design strand. Now, during assay design, the design software looks at probe designs based on both forward and reverse strands. This is because if you report one or the other, you will get the same answer. So here we have an AG SNP on one strand and a TG SNP corresponding on the other strand. We can design probes based on either of these strands. Then the strand designer will choose which of the two will be most likely successful. Now, it bases this assessment on several different things. It bases it on the thermostability of the template to its probe, and it checks also, in this particular example, to see if there are any underlying SNPs. So in this case, we have, for probe design 2, a potential underlying SNP, an AT SNP. And so since there isn't an underlying SNP under probe design 1, our designer would most likely choose probe design 1. So the strand on which the final probe design is based is the Illumina Strand, also known as the design strand. All right? So in the Illumina manifest for BeadChip, you can see here that we have the identifiers for the SNPs on the far left, and we have a name for the assay. And you can see the Illumina Strand and the SNP and the probe sequence. Now, these are all associated. The Illumina Strand, of course, tells us whether the strand is top or bot. So in this column in the manifest, the strand on which the probe was designed has alleles that are top strand alleles for the first assay. For the next one, it's bot strand. What you can see also is that in the SNP column, the SNP column lists the alleles that are based on the Illumina strand. So these alleles are not based on anything to do with the plus or minus strand. They have everything to do with what strand we chose to design the probe on. So the SNP column lists the alleles based on the Illumina strand. All right. So again, Illumina Strand gives the top bot strand designation. The SNP gives allele A and allele B of the SNP on the Illumina Strand. Now note, in this column, the order that the bases are listed in is always genotype A and genotype B. And of course, here we have probe sequences. And you can see sometimes we only have one probe sequence, and at other times we have two probe sequences. That's a topic for another discussion. But we'll move on. Now, there is one more important column associated with the Illumina Strand. This column is called the ref strand. It's placed at the far end of the manifest to the right because it did not used to exist in our original manifest. This ref strand lists the plus or minus strand designation of the Illumina Strand. This is the only reference in the manifest to the genomic or forward strand. So because this refers to the Illumina Strand, this means that all conversions to the plus strand must be compared to the Illumina Strand. So if you have a forward strand and you want to convert it to the plus strand alleles, you have to compare your strand designation that you have to the Illumina Strand. All right? Now, another set of columns here refers to what we have called the source strand. Now, the column source strand gives the top bot designation of the source sequence. The source seq column gives us the sequence of the SNP and its flanking areas as derived from the source. So this is the sequence that we received for design. If it's a DB SNP, then it's the RS entry as listed in the DB SNP database. So if you run a forward strand report, the alleles that are listed here are the alleles that you're going to see. The source seq, again, is the sequence of the SNP and the flanking area derived from the source. Now, if it's a custom chip that manifest we're looking at, and this is a custom design, this is the entry as entered by the customer, you. All right. Now, there's another sequence list here in the manifest that's not extremely useful. This is a column that lists the sequence and the SNP and surrounding sequence based on the Illumina top strand. So these are the alleles from the Illumina top. Many people will confuse this with the plus and the forward, and it's unfortunate that this terminology can be a little confusing. But this is the top genomic sequence based on the Illumina's top strand designation, top bot strand designation. Those are the alleles and strands as listed in the Illumina manifest. The other place where we can find strand designations is Genome Studio Reports. Now, Genome Studio has the capability of outputting the alleles, genotype alleles, based on any strand designation that you wish. A common way of reporting is using the Genome Studio Final Report. Now, the Final Report generates a text report that contains data from fields that you, the user, can select. Now, the Custom Report, on the other hand, outputs files that can be compatible with third-party software. You can download and install plug-in software, small programs, that will create reports that are compatible with third-party analysis software. But we'll look at the Genome Studio Final Report. When you select Final Report and click on Next, you get this window. This window is where you can choose what the report will contain. Now, by default, Illumina created the software and the report by default to report top alleles because at the time it was deemed the most useful strand designation. Now, since the Genome Project has been, Human Genome Project has been completed, most projects, most software that does the analysis is asking for the plus strand or genomic forward strand alleles. You can see here that you can switch the alleles that you choose with other allele formats. You can change the top allele 1 and allele 2 for allele 1 and allele 2 forward, but it's important to note that this forward is the Illumina forward, and as we know, Illumina forward is not equal to genomic forward. If you want your genomic forward alleles, then what you need to do is select allele 1 plus and allele 2 plus. So you can switch these out with the top, and your report will contain the genomic forward or plus strand alleles. Okay? So we now know that Genome Studio outputs reports in various formats. How would we convert data from one strand format to another? It's relatively easy to do if you have Genome Studio. You can simply, as I described, select a report and put it out in a different format. But what if you don't have Genome Studio? Or what if all you have is a flat text report with alleles that you know are from the top strand or Illumina forward strand, and you need the plus strand alleles? Well, in order to transform data from one strand to another, you will need a copy of the CSV version of the manifest. Now, Illumina provides the manifests in a digital format that is used by our software, and it also provides a text version that allows you to open it in a text editor or Excel if it's small enough. So you need to obtain a copy of the CSV or comma separated value version of the manifest and check to see if there is a ref strand column in the manifest. Now, most of our standard chips, the Omni Express, the Omni 2.5, and Omni 5, along with all of our off-the-shelf chips, will have the ref strand in it. Any custom chips or semi-custom chips, the chips which have had content added to them, or chips which are entirely custom, designed by our customers, those will not by default have the ref strand. And so there's if the ref strand is not there, you won't be able to do the transformation using the information in the manifest. Now, if you do have it, you can compare the ref strand data, the plus and minus, Illumina Strand data to which the ref strand plus and minus refers, and the appropriate strand designation that you have. Let's take a look at a simple algorithm that will transform a report that contains the Illumina top alleles to the plus strand alleles. Let's say you have a report, a text report, but the alleles are all reported based on the Illumina top strand, but you want the plus strand. So what we do is we take the manifest and look at some specific columns. In the first example, from your report, if you have your report is as a top strand and you look in the manifest, you see that for a given SNP, the ref strand is plus and the Illumina strand is top. We can use this information to figure out whether we need to keep the alleles in your top report or flip them in order to obtain the plus strand. So you have your top alleles and the Illumina strand is top and the ref strand value in the manifest is plus. You can keep the alleles. Now let's go through that logic. So Illumina strand is top, which means that the strand, the top strand report is the same as your Illumina strand. But we know from the manifest that the Illumina strand is a plus strand. So of course, the top strand and the plus strand alleles are the same, so we can keep them. What if the reference strand is a plus strand in the manifest and the Illumina strand in the manifest is bought? Now in this particular case, we'll need to flip the alleles to the complementary bases. Let's go through the logic of this. So we know that the Illumina strand is bought, but that is the plus strand, which means in your report, which you have the top strand, the alleles are on the minus strand. So if Illumina strand is bought and the ref strand is plus, that means that the top strand allele that you have is on the minus strand, and in order to get the plus strand, you'll need to flip them to the reverse complement. And further example, if the Illumina strand is top, which is the alleles you have in your report, and the ref strand is minus, then clearly to get to the plus strand, we'll need to flip the alleles in your report. And finally, let's say your report is, if the manifest says that the Illumina strand is bought and the ref strand is minus, that means that the bought strand is the minus strand, which by implication means the top strand alleles that you have are on the plus strand, and therefore you can keep these alleles. So here's a simple way to flip a top report to a plus strand report. Use this algorithm by comparing in the manifest the ref strand and Illumina strand values. Okay? Another conversion. Let's say you have the Illumina forward strand report, and you need to convert it to the plus strand. Now in this particular case, you're going to need to actually compare 3 different columns. You're going to need to compare the ref strand, the Illumina strand, and the source strand, which is of course the forward strand according to Illumina. So first, in the first example, we know that the ref strand is plus. That means that the Illumina strand is plus. So Illumina strand is plus, and we have the source strand. Now if the Illumina strand value, top or bot, is the same as the source strand value, top or bot, then we know that Illumina strand and source strand are the same, and Illumina strand is plus, we keep the alleles. If ref strand is plus in the manifest, and the Illumina strand value of top bot is not the same as the source strand, we know that our source strand or forward strand alleles are on the minus strand because it's on the opposite top bot designation. In this particular case, we'll have to flip the alleles in order to get the plus strand. So if the ref strand is the minus strand, that means the Illumina strand is the minus strand, and this Illumina strand top bot designation is the same as the source strand top bot designation. That means that our source strand is on the minus strand, and in order to get the plus alleles, we'll need to flip them. And finally, if the reference strand and the reference strand is minus, that means the Illumina strand is minus, and the Illumina strand does not match the source strand, we know that the Illumina strand is minus, that means, and the source strand is opposite, then that means the source strand is on the plus, and we can keep the alleles. All right? So let's review this conversion of strands. The shortest way to obtain the plus strand alleles is to generate a plus strand final report in Genome Studio, of course. But if you don't have Genome Studio, if you don't have the Genome Studio project, if all you have is a report, then in order to convert them, we'll need to look at the Illumina manifest information. Now, all conversions to plus strand will have to compare the top bot strand designations with the Illumina Strand because the plus and minus determinations in the manifest refer to the Illumina Strand. Now, strand designation clearly has nomenclature that can be confusing. Top, bot, plus, minus, and forward and reverse. They all sound very similar, and unfortunately Illumina's forward and reverse do not match any of those. And that is why we need to use the top bot strand designations to do the conversions. And you need to know what strand designation your downstream analysis tool will require. So let's summarize this. Alternate strand designations were created by different entities who were cataloging genomic variants. Illumina created the top bot strand designation and AB genotype designations as a response to the instability in strand assignments before the assembly of the consensus genome had been completed. Now, top bot designations can be used as a reference to compare and solidify alleles from alternate strand designations. I've included some references here. There are links to the microarray recorded webinars on the Illumina webpage. So there are a lot of webinars that refer to this topic and to many other useful topics covering array analysis. There's also the microarray technical bulletins, and I've listed a few useful ones that are related to the topic we just discussed. The DNA strand designations bulletin, simple guidelines for identifying top and bot strand, the Illumina genotyping manifest column headings, there's a bulletin about that, and how to interpret DNA strand and allele information for Infinium genotyping data. There's also a very old technical note on the website, top bot strand and AB alleles, which was written after we had invented the top bot and AB designations.